Hello students, welcome to EPG Pathshala. I am Dr. Suman Tapriyal from Department of Biotechnology, Central University of Rajasthan. Today I am going to talk about engineering thermostable and cryostable enzymes and site-directed mutagenesis from the paper Molecular Enzymology and Protein Engineering. The objectives of this module are to understand the nature of thermostable enzymes, their industrial applications, the nature of cryostable enzymes and their mechanisms of cryoadaptation, industrial application, biotechnological and molecular biology applications, and protein and engineering of these proteins. Thirdly, we will be discussing about site-directed mutagenesis methods, for example, primer extension methods and PCR-directed methods. Let me introduce to you the thermostable enzymes. Now, these enzymes are actually isolated from thermophiles, the microorganisms living in an extreme high temperature environment. It was around in 1969 that Thomas D. Brook and Hudson Fries discovered a new species of thermophilic bacterium, Thermus aquaticus, in the lower geyser basin of Yellowstone National Park, USA. Now, this discovery started an era of production and characterization of thermostable enzymes. Now, why there was an interest in these thermostable enzymes? Because the microorganisms from where these enzymes were isolated were in thermal equilibrium with their environment, which means that if the organism was able to survive at such a higher temperature, the biochemical reactions were carried out at that temperature, and the biocatalysts, which are enzymes in this respect, must be active, optimally active at that temperature. So the enzymes which were isolated were a subject of study for their stability, for their activity at that higher temperature. So this discovery or this isolation or discovery of these new species of microorganisms paved the way for the engineering of thermostabilizing features in mesophilic enzymes by the use of modern molecular biology and biotechnological use or techniques. Now, as we have come to understand what psychrophilic enzymes and thermophilic enzymes are, let us look at the division of microorganisms on the basis of their growth temperatures. So, let us begin with the psychrophiles. So, the microorganisms which live below 20 degrees Celsius temperature, and this temperature range might go to minus 20 to even minus 60, are called as psychrophiles. Now, the organisms which live within moderate temperatures, which we can call as above 30 degrees and up to 50 degrees, are called as mesophiles. And the organisms which are called thermophiles are actually having their growth temperatures above 50 degrees Celsius. Now, the thermophiles are further classified into thermophiles and hyperthermophiles. So, the organisms which can grow above 50 degrees and up to 80 degrees are called normally as thermophiles, whereas organisms which grow above 80 degrees are called as hyperthermophiles. Now, we will be discussing thermophilic enzymes into a little more detail. So, let us look at the advantages which these thermophilic enzymes provide over mesophilic enzymes. So, these thermophilic enzymes are best suited for harsh industrial processes, as you know, because many of these processes are carried out at temperatures above 100 degrees Celsius. And these thermophilic enzymes can withstand such a higher temperature, and they can show their activity at these temperatures. So, this is the fundamental advantage which is provided by these enzymes. Along with that, these thermophilic enzymes are known to have higher resistance to chemical denaturants as well. The harsh organic solvents and, for example, glutathione hydrochloride, such chemicals which actually leads to denaturation of normal mesophilic enzymes, can be tolerated by these thermophilic enzymes. Another advantage with working with these enzymes is that there is a reduced risk of contamination by common mesophiles. Let's say there is a process going on at higher, more than 100 degrees temperatures. At that temperature, a normal mesophile cannot contaminate the procedure. So this is another advantage of great value. Then the ease of purification is also provided by if you are handling these enzymes in comparison to mesophilic enzymes. So if we are expressing these thermophilic enzymes in mesophilic hosts as we normally do, we will look at that during further slides. These enzymes can be purified from the rest of the mesophilic host enzymes by heat treatment. So the heat treatment can be utilized as a differing characteristic of these enzymes, which can be used for their purification better than other enzymes. Now, thermophilic enzymes allow higher substrate concentrations because we can attain or we can work at higher temperatures. The substrate can be dissolved to a greater concentration at those temperatures, and therefore higher reaction rates are also achieved. So higher temperature leads to a condition which is higher concentration of the substrate, and together can lead to higher reaction rates as well. Now, in addition to these enzymes are known as thermophilic, which show an excellent internal stability for temperature. They serve as very good models for understanding the thermostability and thermoactivity, the structural aspects, the other biochemical aspects of these enzymes, which actually results in the thermostability, and their thermoactivity can be studied using these as models. And the properties which we come to understand because of those studies can be implemented on other mesophilic enzymes where we can introduce such a stability in them. Let's look at the examples. So let me introduce to you the earliest example of a successfully commercialized enzyme which was isolated from Thermus aquaticus, known as TAC polymerase. So this enzyme actually revolutionized the molecular biology techniques because it further led to the discovery of PCR, which is a very base of the molecular biology techniques. So this was the enzyme which was first isolated and commercialized for research activities, which was isolated from a thermophile. Now this enzyme, which is a polymerase, which can amplify or which can make the DNA copies, has an optimal activity at 75 degrees Celsius, and it can withstand a pH of up to 9. Now there further later on different species of such thermophiles led to the isolation of different types of polymerases, and which are now very much used in the today's research. So for example, let us take some more different enzymes. For example, there is a serine protease which was isolated from Thermus strain RT41A. This can be used in DNA and an RNA purification procedures, and it is used in the cellular structure degradation prior to the PCRs. So this protease shows the optimal activity at 90 degrees Celsius, and it can withstand the pH of up to 8. Now the half-life of this enzyme is 20 minutes at 90 degrees Celsius, and plus it requires the use of calcium chloride for its activity. Now there is another enzyme, methionine aminopeptidase. This enzyme we all know is a very important enzyme where it is required to cleave the N-terminal methionine in proteins. And this enzyme is also active at between 85 to 95 degrees Celsius, withstand higher pH as well, and it shows stability of up to 1 hour at 75 degrees Celsius. And there is another kind of enzyme, alkaline phosphatase, which is again used in the molecular biology techniques as the processes where the enzymes are labeled. So the phosphatase at the 5' end is removed by using this enzyme, and this enzyme can also work at a temperature of 85 degrees Celsius at a pH of 9.9. It shows a half-life of 4 hours at 90 degrees Celsius. So as we have seen that how one enzyme can lead to discovery of new methods because of its inherent properties, let us look how these thermophilic enzymes have impacted our industry. So these enzymes, as we have already seen, have an application in the industrial stream. So as it was estimated by a company which is there for market communications, that the global market for industrial enzymes was estimated to be approximately 5 billion at 2013 till 2013, and the expected market for these industrial enzymes is nearly 7 billion. It will grow to this level by 2018 with a compound annual growth rate of 8.2% from 2013 to 2018. Now these commercial figures itself speak about the tremendous implication of these enzymes in our industrial processes. So now let us see how these enzymes are sectioned according to their industrial use. So the industrial enzyme market is separated on 3 broad applications and leads to 3 different sections of these enzymes. So the first section I would like to introduce to you is technical enzymes, where the hydrolases figure into it, and the hydrolases are utilized in starch, food, pharmaceutical, leather, and textile industries. The second very important section of these enzymes is food enzymes. These, for example, chitinase is one of them, and these enzymes are used in food, cosmetic, pharmaceutical, and agrochemical industry. Third section among these enzymes is of animal feed enzymes. The example is phytases. Now these, for example, phytases release phosphorus from the phytate present in, for example, poultry feed. So all these 3 sections combine together and they are serving the industrial requirements to their full capacity. Now among these 3 sections, the largest section is comprised of technical enzymes, and these enzymes, which are hydrolases, are used in detergent, pulp, and paper industry. And this single section of technical enzyme constitute more than 50% of the total world market. The constituent enzymes of these hydrolytic enzymes are proteases and amylases. Now let us take examples of the largest section of these enzymes, the technical enzymes. Let me give you some glimpse about the activity of these enzymes. Let us take amylolytic enzymes, which are comprised of alpha amylases, beta amylases, xylinases. So in an industrial setup, before the use of these hydrolytic enzymes, there is a step of gelatinization of the substrate, which is performed at a very high temperature, and it has to be immediately led into the hydrolytic step. So it can be very easily understood from the requirement of immediate addition of these enzymes at such a high temperature after that step, and therefore these enzymes are very much required, which can work at higher temperatures. So now these enzymes can be isolated from various thermophilic enzymes, from Bacillus subtilis, Bacillus amyloelucifaciens, and Bacillus lechiformis. So there are many producers of these enzymes, and they are isolated, and they can be worked at up to 60 degrees of temperature. The other enzyme which we can look into, the other section, is proteases, which make the second largest leading industrial enzyme. These enzymes are used in food, pharmaceutical, and leather textiles, and the dominant producers of these microorganisms are from the genera Pyrococcus, Thermococcus, and Staphylococcus. So there is an example of extremely stable serine protease, which is actually isolated from a hyperthermophile archaeum, Desulfurococcus. The topic of this slide is enzyme engineering. So now we are moving to the engineering part, which is involving these enzymes. So we all know that for engineering an enzyme, the genes of the enzyme or the subject protein is required in isolation, purified form, where the modifications at the genetic level could be carried out. At the same time, the cloning and expression of these enzymes is performed for their production as well. Now, I would like to introduce one point over here that might arise in your mind, that how is it possible that we are expressing thermophilic enzymes in mesophiles? There is a contrast of their activity and folding pattern between the thermophilic enzymes and mesophiles. So now it has been observed that the thermophilic enzymes can fold up to 60 degrees temperature lower than their growth temperature. And mostly these enzymes can actually be cloned and expressed in mesophiles, where they can be expressed and properly folded in these hosts. So the cloning and expression of these enzymes is going on since 1980s, where the cloning was carried out in mesophilic hosts, which actually provided for the production of these enzymes in large quantities. Now, there were two different methods by which these enzymes were primarily cloned. The first one, where the library of the genes, which was isolated from these host thermophiles, was generated in the mesophilic host, for example, E. coli for a matter, and the selection was done by screening the library for activity. In the second method, an alternate method, it has been seen that the genes were isolated by hybridization or PCR amplification of the gene, followed by their direct cloning into various hosts. To continue into the enzyme engineering and cloning of the thermophilic enzymes, let us see at the various hosts which have been till today been used for commercial production of these enzymes. So there is a range of mesophilic hosts, for example, E. coli, Bacillus subtilis, Saccharomyces cerevisiae, Pichia pastoris, Aspergillus oryzae, K. lactis, and Trichoderma resi. Now, these are the mesophilic hosts which are being generally used for the production of these enzymes. And let us take the example of an alpha amylase. To begin with, alpha amylases, polonases, maltodextrin, phosphorylases, alpha glucosidases, and so on, other enzymes have already been cloned and expressed in these hosts. So the example of an alpha amylase gene from Pyrococcus furiosus was cloned by activity screening, the first method which we talked about earlier in E. coli. And it was reported that the enzyme was isolated with its properly folded structure, which showed its optimal activity at 100 degrees Celsius. The pH range was also maintained between 5.5 to 6, and an advantageous point which was there, and that this enzyme did not require the calcium ions for its activity, which actually was an additional advantage of producing this enzyme in E. coli host. Now we know that the cloning and expression of thermophilic enzymes have been going on since 1980s. So there is an accumulated data, structural data, biochemical data related to these enzymes. So there's a list of enzymes for which the structure of the enzyme has been solved, and a lot of structural features have been found out, which actually led to the thermostability of those enzymes. So let us look at few examples over here which can tell us about the thermostabilizing properties of these enzymes, which can later on be used for modifying other mesophilic enzymes to inculcate the thermostability in them. So as we can see here, ferredoxin enzyme, the structure for which was sold at 1.75 angstrom, it has been shown that there were more charged and neutral hydrogen bonds which were stabilizing the structure of turns in that protein, which contributed to its thermostability. In another example of phosphoglycerate kinase, which was isolated from T. maritime, an organism, it showed that the extension of the helix was there by 4 more residues. So the helical part of the protein was extended. Then there was a dipole stabilization of its C and N caps of the helices. In another example of phosphoribosyl anthranilate isomerase enzyme, the number of prolines was observed to be increased in the loops or at the N-terminus of the helices. So the prolines introduced more at the loops or at the N-terminus led as one of the factors led to the stability of these enzymes. Similarly, the disulfide bridge formed between the enzyme, more number of disulfide bridges led to the higher stability, and it was seen with a pyrrolidone carboxyl peptidase enzyme. With another beta-galactoside enzyme, such increase of ionic pairs where the two ionic side chains of the number of such ion pairs in the protein were found to be increased, and it actually led to the higher stability of this enzyme. Now that we are aware of a number of factors and properties, structural properties, which lead to the thermostability of a thermostable enzyme, there can be an effort to do those modifications in normal enzymes and increase the stability of those enzymes. So let us take one example over here where the thermostability was engineered into an, although it was a thermostable enzyme, it increased the thermostability of that enzyme further. So let us take the example of a protease which was isolated from Bacillus stereothermophilus enzyme, which itself is a thermostable enzyme. However, its thermostability was subjected to mutagenesis, and it was expected to be increasing its thermostability. So the substituents which were introduced into this protease were adapted from an earlier enzyme for which the structure was solved and for which the structural properties were known, which were resulting in the thermostability of the enzyme, thermolysin. So there were 4 substitutions which were done: alanine 4 to threonine, glycine 58 to alanine, threonine 63 to phenylalanine, alanine 69 was substituted by proline. So now, as we can see that these substitutions are actually making the interior or core of the protein more densely packed and more hydrophobic in nature. So that is also an characteristic of the thermostable enzymes. They do have a more compactly packed core with having more higher hydrophobic residues which have higher hydrophobic quotient in them. Now, there was another modification which was done at the residue 65. The serine was substituted for proline. So as we have seen in earlier slide, that the introduction of proline into the structure of a protein results in the thermostability of the protein. And then the another last 2 changes in that protein were done, that a new disulfide bridge was introduced by the introduction of 2 cysteine amino acids. So by doing these changes at the genetic level, the protease, which was already thermostable, was reported to show an increase in its temperature optimum from 74 degrees Celsius to 95 degrees Celsius. Along with that, the half-life of that protein could be increased 300 folds at the 100 degrees Celsius temperature. So this is a very successful example, or a very good example to see that the genetic level modifications in a protein can inculcate more stability, thermostability in an enzyme. And the activity of that mutant enzyme was found to be equivalent to that of the other enzymes from the thermophilic enzymes. So which means we are increasing the thermostability, we are increasing the half-life, but at the same time the activity of the enzyme is not being affected, or rather it is being increased. So students, let's move on to different temperature and the organisms which live on that, which is extremely the other end of the extreme, cryostable enzymes. So the cryostable enzymes are isolated from organisms which are living in the conditions of glaciers or higher, very extreme low temperatures. And as I have already told you that these organisms are living at thermal equilibrium with their environment, which means now, in contrary to thermostable enzymes, cryostable enzymes and the organisms, psychrophiles, which are living in these conditions, are actually in equilibrium with the lower temperature, which can go up to -20, -60. And the biochemical reactions and therefore the activity of the enzymes is related to that temperature. So to begin with the introduction of psychrophiles, which live like in glaciers or other cold regions of the world, the temperature range of these enzymes is 0 degrees but lower than 20 degrees. And as I told already, that the temperature might go to as below as -20 or -60 degrees. Now the important fact about these enzymes is that the activity of these enzymes is characterized by higher turnover numbers, kcat, higher kcat values, and higher catalytic efficiencies, which is kcat by km, and these higher values are reported at low temperatures. So the psychrophilic enzymes or the cryostable enzymes are, as we know, are adapted to cold. So what is the reason behind that? So let us look at that during enzyme reaction, the conversion of an enzyme substrate complex into an activated complex is the step which requires energy. And to generate an active complex, free energy must be added to the enzyme substrate complex. So the magnitude of this free energy of activation, which is actually the energy barrier to the reaction, is reduced in these enzymes. So these enzymes actually target the free energy requirement for changing or converting their enzyme substrate complexes to the activated form, and which actually leads to increased reaction rates. So in normal mesophilic enzymes, if you drop the temperature from 37 to 0 degrees, the activity would drop 20 to 80 times. Whereas if we compare the drop of activity with reference to the drop of temperature in cryostable enzymes, it is not that severely dropped. So the decline of the catalytic activity with the temperature is actually resisted by these cold adapted enzymes. So the activation free energy of psychrophile enzyme, which is the basis of their adaptation to the cold, is contributed by the structural features of these enzymes. So now it is seen that the flexible structure of these cold adapted enzymes allow the reduction in activation enthalpy and allow more negative activation entropies. So these two factors actually leads to lower free energy requirement by these enzymes. So the temperature decrease leads to the rate of reaction of psychrophilic enzymes decrease, but comparatively more slowly. So we have seen that there is a balance of thermodynamic activation parameters which actually contributes to the higher catalytic activity of these enzymes at low temperatures. But collectively, these changes result in concomitant low structural stability also. So there is a downside of this feature of these enzymes, that the enzyme is active in a very narrow range of temperature. So change of that temperature would lead to, because the structure of the enzyme is very flexible, if the temperature of the surrounding reaction or environment is changed above or below that temperature range, the enzyme loses its structural stability to a considerable extent. So by now we know that the catalytic activity of these heat labile cold stable enzymes is correlated to the higher degree of structural flexibility. The heat lability, the word which has been introduced here, actually refer to that small range of temperature in which these enzymes show optimum activity, because their structural stability is lower beyond that range of temperature. So let us look at the examples of one or two enzymes which have structural features contributing to their higher catalytic activity. For example, metalloproteases from psychrophilic bacterium shows the uniform flexible structure where the overall protein of this enzyme is flexible. On the other hand, the enzyme carbonic anhydrase and alpha amylase, which are psychrophilic in nature, they show flexibility in and around the active site, which means the flexibility contributing to the higher catalytic activity of these enzymes is actually located at a specific site. So they show a local flexibility contributing to their higher activity. Cold adaptation of psychrophilic enzymes. As we have seen with thermostable enzymes, there are some structural factors which were contributing to the stability, thermostability, and higher activity of those enzymes. So if we start with, we know that the structural factors, the hydrophobic effects, the main weak interactions which contribute to the protein stability of the enzyme. And these factors may vary in, and rather they do vary in the different types of enzyme isolated from thermophiles or psychrophiles or mesophiles. So in reference to the thermophilic enzymes, if we have to discuss the cold adaptation of the psychrophile enzymes, let us look at the few of these factors which actually contribute to the cold adaptation of these enzymes. So we know, we have seen this earlier, that the proteins of these psychrophilic enzymes are a bit flexible in comparison to their thermophilic counterparts. And it is believed that it is the flexible nature of their three-dimensional structure which actually contributes to their higher catalytic activity at such a low temperature. So there are some structural factors which actually contributes the flexible nature of the antibody for these proteins, enzymes, and which in turn results in their cold adaptation. To begin with, the first is the glycine residues were found to be in more frequency in these enzymes. As we know that glycine amino acid has no side chain, and the polypeptide chain is more flexible on both sides of this residue. So along with that, the higher number of glycine actually introduced higher level of flexibility in these proteins. On the other hand, it was observed that the amino acid which actually inculcates or introduces rigidity into the molecule were reduced in frequency. So the proline, on the other hand, were found to be lesser frequent in these enzymes. Further on, the arginine amino acid was also on a reduction in its appearance. Arginine molecule, as we know, is a positively charged amino acid, is a part of ionic pairing. So the ionic pairing involving arginine actually leads to the stabilization of a protein molecule. On the other hand, we found that a lower number of ion pairs were present and aromatic interactions were also lesser. We know that the core of an antibody or any other molecule or an enzyme for that matter, the core of a protein, if it is compactly packed, it will be more stable. So a smaller size and less compactly packed core will result in the flexible nature of an enzyme. So it was observed that the cyclophilic enzymes, they carry non-polar amino acids which are low on hydrophobicity, and they have comparatively smaller sized core, which again contributes to their flexible nature. Now the structural features can be engineered back and forth in mesophilic and cyclophilic enzymes to increase the enzyme activity and stability respectively, which means if we learn such factors which are contributing to the cold adaptation or thermophilic adaptation can actually be engineered into an enzyme which doesn't show such characteristics. So let us see how these enzymes, cyclophilic enzymes, have found their application in industry and in other research areas. So if we look at the activity of these enzymes, they show higher activity in comparison to mesophilic enzymes, and they show this higher activity at low or moderate temperatures. And it leads, the basic utility of these enzymes is their higher activity at low temperature, which found them industrial applications. So these enzymes are now being used in industries as diverse as molecular biology, even household detergents, biodiesel, bioremediation, and baking, etc. So here are some examples of these cyclophilic enzymes. For example, lipases, proteases, phytases, glucanases, and xylenases. All these hydrolytic enzymes which are cyclophilic, which are active at low temperature, have found application in the animal feed industry, where the use of these enzymes leads to the improvement in the digestibility and assimilation of the animal feed. Then pectinases and xylenases are used in starch hydrolysis at low temperatures. Lipases and proteases, they are being used as in detergents and cleaning industry as additives to the detergents. So these enzymes are added, their properties are adding up to the detergents. Lipases, on the other hand, are being used in biofuel and energy producing industry. They are used to produce biodiesel by trans-esterification of oils and alcohols. Then beta-galactosidases, on the other hand, they are used for production of bioethanol from the dairy waste products. And then chitinases, we know that they are used in pharmaceuticals, medical field for the hydrolysis of chitin to chitosan, chitooligosaccharides, and glucosamine. Now, these cyclophilic enzymes have tremendous application in biotechnological and molecular biology field. So the advantageous feature, I'm going to reiterate that it is high activity at low temperature. And let me introduce one more factor here, is their heat lability. These enzymes can be very easily inactivated by exposing them to mild heat. So the applicability is in the process is performed at or below room temperature. The enzymes are active at low temperatures, and we know that there are many such processes which are carried out in research, which are carried out at a sub-room temperature level. So now, because of the heat liability, it can be efficiently inactivated post-reaction, and we do not require to separate or remove such enzymes prior to the next step in the process. So let us take the example of alkaline phosphatase, which is isolated from a cyclophilic host. So alkaline phosphatase in a normal reaction has to be removed prior to the next ligase or kinase activity. So when we are going to have a ligation activity, or we have to derivatize the DNA by substitution, and we have to use kinases. So these enzymes, ligase and kinase, their activity is actually opposite to alkaline phosphatase. So if you have treated the sample, DNA sample, with alkaline phosphatase, and the next step involves the use of ligase and kinase, in normal procedure the alkaline phosphatase was required to be removed. Now here comes in the heat labile cyclophilic alkaline phosphatase, which has replaced the routine mesophilic enzyme, and this new enzyme allows the next step immediately after the first in the same test tube, because it is heat labile. One can subject the tube or the reaction mix to a small heat and the enzyme can be inactivated, and there is no longer the use or requirement of this enzyme to be separated from the reaction mix. The next step with the ligase and kinase can be subsequently be performed. So the heat labile alkaline phosphatase is in market, and it is marketed by New England Biolab, and it is in very much in use. Let us proceed in this enzyme engineering now. So we know that how these enzymes can be utilized, and we have seen that the activity and stability and their thermal sensitivity of these enzymes can be enhanced to suit their industrial as well as research applications, as we have done with the thermostable enzymes. So after figuring out the stabilizing, cryostabilizing or thermostabilizing features, we know that these features can be introduced into another enzymes of mesophilic origin, and they can be made suitable for a particular reaction. So in this direction, one can use directed evolution technology, which can increase the half-life and activity of coal-adapted enzymes. One such example is lipase B, which was isolated from Candida antarctica, where the half-life of this enzyme after engineering of this enzyme was changed from 8 to 11 minutes, and the activity of the enzyme was shown to be increased 84 per minute to 1900 per minute respectively. So we see that the genetic level modification of these enzymes using the information, structural information to impact the change in their structure can lead to improvement in various factors of or aspects of these enzymes. This shows that higher activity and stability can coexist. Now, this was the earlier generic belief that if you are having a higher activity, the protein will be less stable, or if you are making the protein more stable, you will be compromising at its activity. But this example of lipase B has shown that both of these characters can coexist. So genetic level modification can overcome activity stability trade-off as well. So now these days, along with directed evolution and old empirical mutagenesis methods, there exist now site-directed mutagenesis, where the specific site of the enzyme can be mutated. So for example, Thermophilic subtilase from Bacillus species, such a change in it resulted in 6-fold increase in its casinolytic activity, and it decreased in optimal temperature of activity from 15 to 20 degrees. And the half-life of the enzyme was also increased from 60 seconds to 4 minutes. Site-directed mutagenesis. As we have been discussing so far, that there are characteristics which are native to thermostable enzymes or cryostable enzymes, which can be actually incorporated into mesophilic enzymes which live in moderate temperatures, which show the activity in moderate temperatures and can be modified. Site-directed mutagenesis is a method, or is a technology rather, where you can achieve such a change by substituting one given base or a number of bases in a cloned gene at a desired site. So actually, site-directed mutagenesis gives you a handle where you can, if you have the background knowledge that what and where to mutate a particular amino acid or a structure of a protein, you can actually at the level of genetic molecular level of the gene of that protein, you can bring about those changes. So still now, site-directed mutagenesis have been very aptly employed in structure function related studies, where the gene of a protein generally is being mutated at random positions to check, or the specific positions to check whether which amino acid contributes to its structure or which amino acid contributes to its activity. Along with this application, the site-directed mutagenesis is also used for the production of beneficial mutants with novel properties. Just like we can generate we can incorporate the thermostability characteristics in a mesophilic enzyme, and we can produce a mutant of its original wild type, which could be used in industry or clinical or therapeutic fields. So the site-directed mutagenesis, as I told, is a protocol, is a technique, can be achieved via different methods. There are methods which were earlier used, which are lengthy and laborsome methods, but still in use, and that is primer extension and its variation gene editor method. Nowadays, a more prevalent method which is being used is PCR-based classical method, and its variants, simple methods, egg site method and gene tailored method. Let us look at the primer extension method. This method involves in vitro synthesis of the target DNA using a synthetic oligonucleotide. This is also known as primer extension experiment. As you can see on the slide itself, that the primer is carrying the mutation in it. So this method actually basically requires the target gene to be changed in the single stranded form. Nowadays even the double stranded form can be seen. We will look into that after some time. So the mutations of addition or deletion or a point mutation at one single base or a couple of bases can be introduced into the primer, and the primer can be used to synthesize a DNA which will harbor that mutation in the subsequent progeny molecules. So in the primer extension method, let us look little more deeper how this method works. So this is as I told, an oligonucleotide directed mutagenesis. The primer which is used in this method is actually harboring the mutation in it. So generally you would require a single stranded target gene, which you can generate by cloning your DNA into M13 vector. So the primer and the target genes are mixed together, and the reaction is performed in the presence of DNA polymerase, DNTPs, T4 DNA ligases, and ATP. And then as you see that it results into the synthesis of the double-stranded DNA. Now this is an heteroduplex where one of the strand is the original strand, the target which is carrying the unmutated target gene, and the second newly synthesized strand carries the mutation in it. Now this mix which is carrying this can be now transformed into E. coli host, and then the E. coli host which is now going to multiply these heteroduplexes further into homoduplexes and the original homoduplexes can be screened off those colonies by screening methods, by hybridization screening methods. So once the heteroduplex which was synthesized in a test tube has been transformed into the E. coli, the further multiplication by the E. coli takes care of converting the heteroduplexes into both the types of the DNA strands. So some of the bacteria will produce the double-stranded DNA with the homoduplex DNA, which is the completely mutated form, and it will also lead to the regeneration or double duplex formation of the original gene. So now once this has been carried out, one has to further screen the colony for the isolation using the stringent methods for the isolation of the clones which are harboring the mutated forms. So now let us move to a more popular primer extension method. As I said that the earlier methods which oligonucleotide directed mutagenizing primer extension methods were simple, but they were having few disadvantages to them. For example, the procedure or the reaction mix was contaminated with single-stranded, non-mutated templates or even partially double-stranded molecules. Although these other molecules can be separated by the use of agarose gel electrophoresis, the process becomes laborious and lengthy. On the other hand, using this content of DNA reaction mix, you would require a mutant host which is being made deficient in methyl-directed repair of mismatches, because in a normal host, such a small stop mutagenesis is generally corrected by DNA methyl directed mismatch repair system of the E. coli. So with all these complications nowadays, to overcome these, commercially available mutagenizing kits are available which use multiple primers, not a single primer. They use more than one primer, and they are generally using nowadays double stranded templates as well. So let us take a look at the gene editor system here for generating mutations using site-directed mutagenesis. As you see on your screen slide, that there are two primers. One is mutagenesis primer 1, and then there is a resistance capacity enhancing primer 2. As you all know that for the selection of a recombinant plasmid DNA, there is always a selection gene which is generally a antibiotic resistance gene. So the primer mutagenesis primer 1 is the primer which is actually carrying the mutation in it, whereas the resistant capacity enhancing primer has the mutation in it which would actually lead to the enhancement of the resistance against the antibiotic. So both the primers here are performing their work. So we can use a single stranded DNA, or the double stranded DNA can be used at the same time, but these primers, mutagenizing primers and the resistance enhancing primer 2, will perform their activity in the presence of DNA polymerase, DNA 4 ligase, ATP, DNTPs, as we have seen in the earlier example. Once this reaction has been completed, this heteroduplex is again transformed into the E. coli host. And now again, once the E. coli host has been transformed with this DNA heteroduplex, would further lead to the generation of double-stranded homoduplexes of the mutated target as well as the wild-type target. But here we can see that the mutated double-stranded fragment has a mutated resistance gene. So the selection of the recombinant which has the desired mutation, it can be selected at a slightly higher resistance, at a slightly higher concentration of the antibiotic, for which the resistance has been now incorporated into the mutated duplex. Let us look at another type of mutagenesis method, which is PCR based. So now we are going to employ the PCR as one of the methods in this, and the method we are going to discuss is excite method. Now the difference, the very basic difference here is that the target DNA which is going to be subjected to mutagenesis have to be generated in a host which has its restriction modification system intact, which means it would produce DNA molecules which will be methylated. So that is the prerequisite of this method. So once we amplify the recombinant DNA which has to be mutagenized in a host, in such a host which would make the recombinant plasmid methylated, an isolation of this target DNA can then be subjected to the mutagenesis method. So this method also uses two non-overlapping primers. The position of that, as you can see on the slide, they are spanning the target gene. Now only one of this primer carries the mutation. So there is one mutagenesis primer 1, and there is a reverse primer. And the target gene, as we can see, is double-stranded here. Now this is subjected to few cycles of PCR. So the use of these mutagenesis primer 1 and reverse primer would generate a linear fragment, amplification of a linear fragment. And as we know that the reaction is carried out in vitro, the newly synthesized DNA which is harboring the mutation at its 5' end is non-methylated. Along with that, you would be having contaminating original circular DNA which is wild-type DNA. Now if we look at the stage at which we are here, the simple PCR has amplified a DNA which is mutagenized from a target DNA. So we have our mutated DNA in linear form with us here. So now we are going to subject this PCR amplified reaction mix with an enzyme which can target only the methylated sites. So the mixture of this PCR amplification is subjected to the DPN restriction. Now DPN1 will target only the contaminating original circular DNA which is methylated, because it can target only the methylated DNA, and our linear DNA will not be affected by this enzymatic treatment. Now the linear DNA can be purified from this mix and can be subjected to a ligase where the DNA takes its again the original covalently close form as a plasmid, and then after this treatment the recombinant plasmid harboring the mutation in it can be retransformed into another fresh E. coli host for isolation. So students, let us now summarize what we have learned in this module. To begin with, these thermostable and cryostable enzymes have found applications in industrial processes, therapeutics, and household products. For example, thermostable hydrolyses are there, which are being used in starch, food, pharmaceutical, leather and textile industries. Then cryostable lipases, proteases, phytases, glucanases, and xylenases are used with animal feed. Secondly, if we see the thermostable and cryostable stabilizing straits can be introduced at genetic level to modify the amino acid sequence of a target mesophilic enzymes. Similarly, enzymes isolated from thermophiles and psychrophiles can be modified to attain substrate specificity and activity of a mesophilic enzyme. If we move further and look into that, the advanced PCR-based mutagenesis methods, for example, X-site and gene tailor, have become routine tools to modify proteins of research, diagnostics, and therapeutic applications. Thank you.